Welcome to our channel, Mootechie. Today's topic, classification of viruses. Here we see a visual overview of the three major themes we'll be covering today: nucleic acid, morphology, and the Baltimore system. Lecture overview, classification of viruses. First, we'll define the key classification bases and criteria used in virology. Then we'll learn to recognize major morphologies and whether a virus has an envelope. We'll compare DNA versus RNA categories and their structural features. Finally, we'll explain the Baltimore system and, very importantly, its clinical relevance. Understanding why we classify viruses this way is just as crucial as knowing the categories themselves. Basis of viral classification. These are the 5 fundamental criteria used to categorize viruses. First is nucleic acid. This includes whether it's DNA or RNA, its sense, if it's single or double-stranded, and if the genome is segmented. Second is morphology. This looks at the virus particle's physical structure, its capsid symmetry, its overall shape, the presence of an envelope, and surface spikes. Third is replication strategy. This dives into the molecular details, what polymerases the virus uses, its transcription pathway, and where it assembles inside the host cell. Fourth is host range and tropism. This asks what does it infect, animals, plants, or bacteria? And within a host, does it prefer specific tissues or cell types? Fifth is genome organization. This considers the size of the genome, whether it's linear or circular, and if it's segmented into multiple pieces or is one continuous strand. These 5 pillars together give us a comprehensive picture of any virus. Classification by nucleic acid. This is the primary division in viral taxonomy, the very composition of its genome. Let's start with DNA viruses. These are generally stable and have error-correcting mechanisms. A major group is double-stranded DNA or dsDNA. This includes most DNA viruses. They typically replicate in the host cell's nucleus except for pox viruses. Examples include herpes viridae, adenoviridae, and pox viridae. Another group is single-stranded DNA or ssDNA. These viruses are simpler but require the host cell to first synthesize a complementary strand to form double-stranded DNA before they can replicate. An example is parvoviridae. Key characteristics of DNA viruses include high genetic stability due to proofreading enzymes, and they often establish latent or persistent infections in the host. Now let's look at RNA viruses. This group is incredibly diverse and known for rapidly mutating. First, positive-sense single-stranded RNA or plus ssRNA. For these viruses, their genome can act directly as messenger RNA, which means the viral particle itself is infectious. Examples include picornaviridae, which causes polio, and coronaviridae. Next is negative-sense single-stranded RNA or minus ssRNA. Their genome is the complement of mRNA, so they must carry their own viral polymerase enzyme into the host cell to make a proper mRNA strand. Examples are orthomyxoviridae, like influenza, and rhabdoviridae, like rabies. There's also double-stranded RNA or dsRNA. An important example is reoviridae, which includes rotavirus. Key characteristics of RNA viruses include high mutation rates, which lead to phenomena like antigenic drift in flu viruses, and they mostly replicate in the host cell cytoplasm, not the nucleus. Classification by morphology. This is categorization based on the virus's capsid symmetry and overall structure. From Mootechie. There are 3 main structural types: icosahedral, helical, and complex. Icosahedral viruses have a symmetry based on 20 equilateral triangular faces. This is nature's most efficient way to enclose space with identical subunits. They appear roughly spherical under a microscope. Their building blocks, called capsomeres, are arranged in precise geometric patterns, often as pentons and hexons. Examples include adenovirus, poliovirus, and herpes virus. Helical viruses have their capsomeres stacked in a spiral around a central axis, like a spiral staircase. This gives them a rod-shaped or filamentous appearance. An important note, most human-infecting helical viruses are also enveloped, meaning they have a lipid membrane stolen from the host cell. Examples are influenza, rabies, and the classic plant virus, tobacco mosaic virus. Complex viruses don't fit neatly into the other two categories. They have neither purely icosahedral nor helical symmetry. Their structure is often multi-layered, or they possess extra features like tails or outer walls. They are generally larger viruses. Prime examples are pox viruses, which have a unique brick-shaped appearance, and bacteriophages, the viruses that infect bacteria, which often have intricate tail structures. Baltimore Classification. This is a powerful system that groups viruses into 7 classes based on their pathway to synthesize messenger RNA. Remember, mRNA is the essential molecule that tells the cell how to make viral proteins. Class 1 is double-stranded DNA viruses. Examples are herpes and pox. Class 2 is single-stranded DNA viruses, like parvovirus. Class 3 is double-stranded RNA viruses, such as reovirus. Class 4 is positive-sense single-stranded RNA viruses, including poliovirus and coronavirus. Class 5 is negative-sense single-stranded RNA viruses, influenza and rabies. Class 6 is single-stranded RNA retroviruses, like HIV. They use reverse transcriptase to make a DNA copy of their RNA genome. Class 7 is double-stranded DNA viruses that also use reverse transcriptase, like hepatitis B virus. The key concept to lock in is this: all viruses must eventually generate a positive-sense mRNA molecule to produce their proteins. The Baltimore system brilliantly groups them by how they get from their starting genome to that crucial mRNA. Importance of viral classification. Now, why does this systematic categorization matter so much in clinical medicine and research? First, it guides diagnosis and treatment. Accurate identification of a virus helps clinicians choose the right antiviral drugs and formulate effective patient management plans. Second, it predicts transmission and epidemiology. Knowing a virus's class can tell us about its environmental stability, its likely routes of spread, and even potential outbreak patterns. Third, it informs vaccine design. Understanding a virus's surface structures and how it replicates is absolutely fundamental to creating safe and effective vaccines. Fourth, it standardizes nomenclature. It gives researchers and doctors around the world a common language to discuss viruses clearly, avoiding confusion. Finally, it prioritizes biosafety. A virus's classification helps determine the necessary containment level in a lab, from biosafety level 1 all the way up to the maximum containment of BSL-4, ensuring safe handling protocols. Let's quickly recap the core pillars, the 5 bases of classification, like nucleic acid and morphology. We compared genome types, stable DNA versus highly mutable RNA. We explored the 3 main physical structures, icosahedral, helical, and complex, and we decoded the Baltimore system with its 7 classes, all centered around mRNA pathway. Here's an exam pro tip. Always link the Baltimore class to the replication enzyme required. For example, remember that class 5 viruses need an RNA-dependent RNA polymerase. Examiners love to ask, where does the mRNA come from, and now you know how to trace that path. You've done excellent work building this strong foundation in virology. Thank you.